In this video tutorial, we will cover the use of filters and the application of post-processing information in the specimen management module of Web LDMS. In order to find specimens, we utilize filters, and those filters are located in a panel to the left-hand side of the screen. They are separated out by categories. Our first category is participant filters. Here you can set a filter on your project, your ID1 or participant identifier, or the other participant identifier if used. The second category is enrollment filters, where you will set your study or protocol. The third category is visit filters. Here you can set a visit value, visit unit, clinic, ID3. For ACTG impact, this is the study identifier. Use of this field varies by network. The last option here is collection date. Now this is a quick filter that you can put in place to find all the specimens that were collected at one time. Our next category is specimen filters. Here you can filter on the specimen identifiers, such as Global Specimen ID, the LDMS Specimen ID if you've migrated over from our Windows system, and the other Specimen ID if used. The Global Specimen ID is the most restrictive filter you can place, since a Global Specimen ID only belongs to one specimen at a time. When this filter is active, only one specimen will be shown, and all other data in specimen management will be hidden. You can filter on primary type, additive, derivative, and subadditive derivative type. You can filter by a particular technologist initials to see any of the entries that they have made. At the bottom of this category, you see the received date. And this, along with collection date, very powerful and quick filters you can use to find all the specimens from one date in order to adjust the information that you've entered. Our next category is shipment filters. You can filter by batch number, the date that you imported a shipping file, an intended ship date if specimens are on a batch. Our storage filters. Here you can filter by a particular storage item, such as a freezer, a rack, or container, to find all the specimens that are contained within that storage item. Now let's apply some specimen filters in order to find viable PBMCs and add our processing information. The first filter I'd like to apply is a collection date. I'll go to collection date field, click the down button, a calendar will appear, and I will set the date. Click add to apply the filter, and we already see in our specimen management screen that we have one participant in our participant counter and one visit for that participant, which matches that collection date. If you had multiple participants on that date, then you would have more records that you could go to. We can apply a specimen filter in order to find our viable PBMCs. Those are coded in the LDMS as cells with a derivative of CEL. Click to apply the filter. And now with those two filters set, we see all of the viable PBMCs that were collected on that particular date. In order to add the processing information, we will start in the primary edit menu. Clicking edit will bring up our pop-up menu, and scrolling down we will see our specimen processing details section, starting with our processing tech initials. Our processing date, which defaults to the received date but can be adjusted if necessary, and our processing time. We also have our total cell count. Once you've added this information, click the Save button. You're asked if you wish to cascade the information that you've added to the aliquots. By clicking yes, that information will then be applied at the aliquot level. To enter information for our aliquots, we can enter each edit button individually by simply clicking Edit, scrolling down. Below our specimen processing detail section is our frozen date and frozen time. The frozen date defaults to the processing date but can be adjusted, and I have entered in our frozen time. I'll click the Save button in the bottom center of the menu, and now the frozen date and time has been updated for that one aliquot. To edit multiple aliquots at once, click the first aliquot, hold the Shift key, and click the last aliquot. Move your mouse to click one of the edit menus. And here we see a different type of edit menu. A lot of the information is present here that we saw in the first aliquot edit menu. But in order to edit the information, you must first click the checkbox to the left of the field. The field will open up, and then you can enter in the information. Click Save, and it will be applied to all selected aliquots. Thank you for taking the time to watch this video. Further information can be found in the LDMS user manual on the LDMS website at www.ldms.org. You may feel free to contact LDMS user support. The Contact Us information is located on the website as well.